Okay, our next speaker is Dr. Nathanael Gray. Dr. Gray is a chemist, and we were thrilled when we were able to recruit him to Stanford in general, and our lung cancer program in particular. Dr. Gray has made many very important contributions in the area of drug development for lung cancer, particularly for non-smoking lung cancer, with major contributions for developing inhibitors for ALK mutations as well as EGFR mutations. I'm sure he'll be talking about. Please welcome Dr. Gray.
Great. So thank you so much for the organizers for the opportunity to speak here, and apologize for being a chemist. You know, chemistry is the subject used to torture doctors and try to prevent them from becoming doctors. So then when you tell people you're a chemist, they're often, disappointed. But our goal here really is to, figure out new strategies for targeting EGFR. How do we come up with the next generation compounds? And my message is really, very simple. We're trying to innovate, new approaches for harnessing these mutations such that when resistance arises to osimertinib, there will be future options. So I think everyone is well familiar with this drug discovery story. Actually, when this project started in lung cancer, we were targeting overexpressed, EGFR, and it was from seminal work here and in Boston that we really figured out that only patients, that harbored these specific mutations were sensitive. And then I think the other breakthrough came from work that we led early on, convincing people that you could make covalent kinase inhibitors. So at the time, we started this work in the early 2000s. People didn't really believe in covalent drugs. But we realized that there is a unique cysteine residue in EGFR where you can actually form a covalent bond. So most drugs, they bind and unbind. This drug forms a permanent covalent linkage to that cysteine. And so that early work developing this tool compound ultimately led AstraZeneca to develop osimertinib, which is now the mainstay drug. And of course, as time has evolved, we're beginning to learn about, what are the features of these compounds that make them favorable. One of the key features was, for example, overcoming this first mutation, this T790M mutation, which gave resistance to the first-generation drugs. The other problem that we were trying to overcome was the first-generation drugs gave a very nasty skin rash and diarrhea. And that turns out to come from wild-type EGFR inhibition. And so we figured out that by covalent modification, we could make them more mutant selective and better tolerated. So finding compounds that can circumvent resistance mutations while still being very mutant selective is what led to better and better compounds. So I think all of you are familiar with the overall drug discovery process from the discovery stage to preclinical, clinical research, and eventually to approval and regulatory compliance. One of the real challenges here is, the disciplines that are required are very diverse, and the process can be quite inefficient because you have very different stakeholders, in the process as you move this through. And so one thing we've been trying to do at Stanford is come through with more efficient processes where we can go from research into approval. And I think you've seen the benefits of this now with the ALK inhibitors, where between the time that EML4 ALK was found as a driver in lung cancer to when those drugs were approved only took 5 or 6 years. Whereas for EGFR, it was almost a 15-year journey before we had approved compounds. So one of the things we're focused on right now is the C797S mutation. Remember I told you that osimertinib is a covalent drug. So as you would expect, you put the tumor under incredible selective pressure to try to escape that drug. And one of the easiest ways for the tumor to escape that drug is by changing the residue that the drug binds onto, so that cysteine gets changed to a serine residue, and that gives resistance. So together with my colleague Michael Eck, we figured out that there's another binding pocket in EGFR called an allosteric binding pocket, here shown in green, away from the ATP binding site. And so we reasoned if we could find a second site binder, that would be one way to circumvent this cysteine mutation. And so we did a screen at Dana-Farber about 10 years ago, and we found a very good allosteric binder. We did a lot of medicinal chemistry, and we optimized it. And then eventually, we got the compound into a startup company actually in China, and this is a disclosure, I'm involved with this company. This company was now optimized this compound to a drug candidate, and then they partnered actually with AstraZeneca. And so now AstraZeneca is soon to bring this compound to the clinic, and we're really excited about this because this drug can co-bind with osimertinib. So you can keep taking osimertinib, and then you can add this additional drug on there, and 2 drugs is better than 1 drug, and it makes it much harder for the tumor to escape because you are now contacting 2 different surfaces. A second approach that we've also pioneered is a degradation approach. So instead of just inhibiting EGFR, why don't we try to completely eliminate it? And it turns out our cells have a very efficient elimination process for proteins called the proteasome. It's basically the garbage disposal for the cell. And a whole bunch of researchers over the last decade have figured out how do we tag proteins for degradation by this garbage disposal. And we figured out the chemistry strategies for basically making a molecular glue that then tells the garbage disposal to grab onto that protein and degrade it. And so Jaebong and the lab developed the first degraders of EGFR. We then helped start a startup company in Boston called C4 Therapeutics. They took our academic tool compound, they polished it further, they solved a lot of problems. This compound is a very good degrader of EGFR. They showed it worked in all the preclinical models, and then they actually partnered with a company in China, and now this compound is undergoing clinical trials in China in a subset of EGFR mutant lung cancer. And we're hoping, if the data there is positive, that we'll also be able to launch a clinical trial in the US. So this will be the first EGFR degradation strategy in comparing inhibition versus degradation. So here's sort of the pipeline that we've created over the last 20 years, all targeting EGFR. The 3 that I mentioned are now approaching clinical trials. And in the last 8 minutes, I'll tell you about what's on the horizon beyond direct EGFR inhibitors. So we have 2 approaches that we're actively working on in the lab right now. One is called a molecular bidentate, and the other is called a proximity induction. So that sounds highfalutin, but the basic idea is extremely simple. So for the molecular bidentates, we realized that there's actually 2 cysteine residues in the ATP site. So osimertinib grabs onto this one. But we're like, why don't we also grab onto this one over here, right? When you're rock climbing, it's always good to have 2 hands on the rock and not 1 hand on the rock. So this is actually a much more powerful way of suppressing EGFR activity because you're actually cross-linking the enzyme. So here, what we basically have been able to do is have a compound that will be impervious to just a single mutation. So as I mentioned, osimertinib, you can progress if you get the C797S mutation. And so we simply gave the compound another hand to grab onto this other residue. And so we did it, for example, with this compound. This part of the molecule grabs onto the front. This part of the molecule grabs onto the back. And then we were able to show in the lab that the only way EGFR can now escape from this molecule is by mutating both of these residues. And that's statistically much harder for the tumor to do. And so right now we're doing preclinical work to convince folks that this molecule is more difficult to get resistance to. And here's sort of a classical study where you select tumor cells with the inhibitor, and we ask, how many resistant clones do you get? And when you do get resistant clones, where are the mutations? And these studies are very promising. So you can see here, osimertinib in culture, you can get resistance actually quite quickly. This one is much harder to get resistance to, and now we also have to show this in animal models as well. So that's the molecular bidentate approach. What about this other approach, induced proximity pharmacology? So I taught you, I'm going to go about degradation. So degradation is induced proximity to the proteasome, so you're basically taking the protein to the garbage disposal and then degrading it, what else can you bring EGFR in proximity to? So here the idea is that instead of inhibiting EGFR, we're going to use EGFR as a kill switch. So basically, we're going to have a molecule that if you're mutant EGFR, it's going to tell that cell to commit suicide. And so the trick is to find another protein that when glued to EGFR will induce apoptosis, necroptosis, and ferroptosis. So you're basically making a kill switch. So we're looking now at a variety of ways of achieving this. It turns out our bodies do this quite naturally. We have a cellular process called apoptosis that eliminates billions of cells every day in our body. And so we need to figure out how to subvert that apoptosis process with mutant EGFR. And so we have several proteins now where we're making this molecular glue, and then we want to show that this active way of killing cells will have less resistance than simply inhibiting EGFR, right? So inhibiting EGFR, you're killing the oncogenic driver. With this method, you're actively inducing cell death, and we hope that'll lead to a very deeper kill of the cell population and not leave around these drug-tolerant persisters where a lot of the resistant clones arise. So one thing that's really key in this drug discovery process is you need very diverse expertise to work on this. And so everything that I've been able to achieve in EGFR over the last 20 years is because of these three people. And so Pasi, who's a physician scientist at the Farber, really pulled us into the EGFR arena. And then Michael Eck is the world leader in structural biology, right? So you can't understand what you can't see. And so he's been able to get crystal structures of EGFR, inform our design of compounds. And then Kwok Wang had been a leader in modeling EGFR. So how do you convince people that you've made a good drug? Well, you have to show that it works in an in vivo model, and he developed those models. I also want to point out we've got a really amazing team, EGFR team now assembled at Stanford, really focused on these new EGFR approaches. And I also want to point out that our funding right now is really being challenged. Clearly we have to convince taxpayers that EGFR and lung cancer remains a big problem, but we also have to do a better job of establishing philanthropic support, working with foundations, and partnering with companies to keep this productive ecosystem going. And as I mentioned, there's lots of different stakeholders here, patient advocates, companies that want to make money, taxpayers that want to know their money is going to a good end. And I think we as a community really have to work towards keeping all the stakeholders engaged. So also want to thank an amazing research team we have at Stanford. These are the real heroes of the work who go in the lab every day and try to tackle EGFR. So I'll stop there, and I will turn it over to the next speaker. Thanks.